We've been searching for the secrets of aging for thousands of years, but scientists are finally beginning to unravel the mysteries of how and why we age. Some of the answers to these questions can be found in our DNA, in structures called telomeres. Telomeres are protective caps found at the ends of our chromosomes. They are thought to play a major role in aging by limiting the number of times most of the cells in our body can divide. They help protect our genome and defend our bodies from developing cancer. That protection is related to the number of DNA repeats in the telomere itself. Ours can have anywhere between a few hundred to a few thousand repeats, all with the same distinctive DNA sequence. TTAGGG. The repeats bind a set of proteins called Shelterin, and together they protect the DNA by forming a closed telomere cap. But every time our cells divide, DNA repeats are lost, so our telomeres shorten, like a slowly burning candle. Too short or uncapped telomeres signal to the cell that it's time to retire. The retiring cells stay in a resting state called senescence, meaning they stop dividing, or they undergo programmed cell death. Most cell divisions take place during development in the first few years of life, but telomere length continues to decline in most cells at a steady rate as we age. So it may seem like shortening telomeres are simply dangerous, but this burning candle is actually a critical part of how our bodies prevent cancer.
When telomeres are too short, that's an indicator that cells have accumulated too much damage or have divided too many times, possibly long enough to have acquired dangerous mutations. So telomere loss is an important mechanism to suppress tumor growth.
But what about our cells that need to be able to keep dividing? Some cells, like stem cells, need to divide many times over our lifetime to replenish tissues and maintain their function as we age. But even stem cell telomeres aren't immune to shortening. That's where telomerase comes in. It's an enzyme that lengthens telomeres. Not enough to completely prevent shortening in most cells of our body, but enough to slow it significantly. Telomerase is particularly active during early-stage embryonic development and in the stem cells of the germline that produce sperm and eggs. This means these cells are not limited by telomere shortening to the same degree other cells are. It may seem like pumping all cells in our body full of telomerase would be a good idea to prevent telomere shortening, and along with it, aging itself. But since we'd be bypassing our body's cancer security mechanism, that could be disastrous. Telomere shortening stops cells from replicating endlessly. When cells do replicate endlessly, they become cancerous. Sitting in a unique place at the intersection of cancer and aging, there's good reason for scientists increasing focus on telomere research. And as research continues, we may yet discover the answers to some of the most enduring mysteries in science. This is the first in a short series of videos where we explore telomeres and the impacts they have on our health. In the next episode, we'll take a look at why telomeres shorten over time.